Familiarization with laboratory equipment and basic laboratory techniques for field crop physiology. Analytical balance. Used to accurately measure small quantities of plant materials and chemical substances, ensuring precision in experimental work. It is essential for determining the fresh and dry weights of plant samples to assess biomass accumulation, growth rate, and moisture content. The balance is also used in preparing chemical solutions, weighing reagents, and quantifying pigments or nutrients such as chlorophyll and nitrogen. Additionally, it helps measure seed or grain weight and ensures accurate application of growth regulators and treatments. Overall, the analytical balance plays a vital role in obtaining reliable and precise data for studying various physiological processes. pH meter. Used to measure the acidity or alkalinity of soil, water, and nutrient solutions, which greatly affects plant growth and physiological processes. It helps determine the optimal pH range for nutrient availability and absorption by plant roots. Researchers use the pH meter to monitor and adjust the pH of culture media, irrigation water, and hydroponic solutions to maintain suitable conditions for plant development. It is also used in experiments analyzing how different pH levels influence enzyme activity, photosynthesis, and overall plant metabolism. Overall, the pH meter is an essential tool in crop physiology for ensuring accurate measurement and control of the chemical environment that supports healthy and efficient crop growth. Electric compound microscope. Used to observe and study the detailed structure of plant cells and tissues that are not visible to the naked eye. It allows researchers to examine cell organelles, stomata, chloroplasts, xylem and phloem tissues, and other microscopic features that play vital roles in physiological processes such as photosynthesis, transpiration, and nutrient transport. The microscope helps in identifying cellular changes caused by environmental stress, diseases, or treatments applied to the plant. It is also used in studying plant anatomy and understanding how cellular structures contribute to overall plant function. Overall, the electric compound microscope is an essential tool in crop physiology for analyzing the internal organization and physiological condition of plant tissues at the microscopic level. Hot plate stirrer. Used to heat and mix solutions evenly during the preparation of chemical reagents, nutrient media, and plant extract samples. It ensures uniform temperature and proper dissolution of substances, which is essential for accurate experimental results. The stirring function helps maintain homogeneity in solutions such as fertilizers, growth regulators, or buffer solutions used in physiological and biochemical analyses. It is also used in procedures that require controlled heating, such as chlorophyll extraction or enzyme activity assays. Overall, the hot plate stirrer is an important laboratory tool in crop physiology that provides consistent heating and mixing, ensuring the reliability and precision of experimental preparations and analysis. Microwave plasma atomic emission spectroscopy. Used to analyze and quantify the mineral and nutrient content of plant tissues and soil samples. It provides precise measurements of essential elements such as nitrogen, phosphorus, potassium, calcium, magnesium, and trace micronutrients that influence plant growth and physiological functions. This instrument works by exciting atoms in a microwave plasma, causing them to emit light at specific wavelengths that correspond to different elements. Researchers use MPAs to study nutrient uptake, deficiency, and accumulation in plants under various environmental or treatment conditions. Overall, MPAs is a powerful analytical tool in crop physiology that helps ensure accurate evaluation of plant nutrition and support studies on nutrient management and soil-plant relationships. Electric weighing scale. Used to quickly and accurately measure the weight of plant samples, soil, fertilizers, and other materials needed in experiments. It is especially useful for determining the fresh and dry weights of plant parts, such as leaves, stems, roots, and fruits, to assess growth performance and biomass production. The electric weighing scale provides precise and consistent readings, which help reduce human error during data collection. It is also used in preparing nutrient solutions, weighing harvested samples, and monitoring plant yield in physiological studies. Overall, the electric weighing scale is an essential tool in crop physiology for ensuring accuracy, efficiency, and reliability in measuring plant and experimental materials. Laboratory over. Used to dry plant samples, such as leaves, stems, and roots, at controlled temperatures usually between 60 degrees Celsius to 80 degrees Celsius, until they reach a constant weight. This process removes moisture from the samples, allowing researchers to determine the dry weight, which is essential for calculating biomass, moisture content, and growth rate. It is also used to dry soil samples, sterilize glassware, and prepare samples for further chemical or nutrient analysis. Familiarization with laboratory equipment and basic laboratory techniques is important in field crop physiology because it ensures accuracy, reliability, and safety in conducting experiments and analyzing plant samples. Understanding how to properly use equipment such as balances, pH meters, ovens, and microscopes helps researchers obtain precise measurements and valid results when studying plant growth, nutrient content, and physiological responses. It also promotes efficiency in sample preparation, data collection, and analysis, reducing errors that could affect the interpretation of results. Moreover, being skilled in basic laboratory techniques enhances the ability to apply theoretical knowledge to practical situations, linking laboratory findings to real field conditions. Overall, familiarity with laboratory tools and methods is essential for developing scientific competence and achieving meaningful outcomes in crop physiology research.